Hello, viewers. Welcome to Biodrunks. I'll be providing notes, PDF link in the description.
In the previous video, we learned about gene cloning, that is genomic library construction. So while doing that, we came across a point called vector. Let's learn more detailed about it. So what are vectors? Vector is a DNA molecule that has an ability to replicate autonomously in an appropriate host. Okay, so what happens is in that vector we have the target DNA inserted. So carry a target DNA, that means a target DNA is inserted into it. It can be used for either cloning or expression. Okay, now how see vectors, they should have an origin of DNA replication, which is also known as ORI, that functions in the host cell. So any extra chromosomal small genome like plasmid, phage or virus can be used as a vector. What are the properties of a vector? Like what do we look at a vector when we have to use it? The first and foremost property is it should replicate autonomously. So here is the objective of cloning. What is the objective of cloning? We have to obtain large number of copies. Okay, so I need large number of copies. So when I need large number of copies, it should be very much clear that the vector should be replicating autonomously. Okay, which will be giving me multiple copies. The size should be less than 10 kb. See, because large DNA molecules are broken during the purification procedure. And in addition, there are large vectors that present difficulties during various manipulations while gene cloning. So we always prefer having a vector with a small size. It should be easy to isolate and purify. Yes, this is most important and the vital step. If you're not isolating the vector properly or you're not purifying it properly, it would cause a problem in further gene cloning experiments. It should be easily introduced into the host cell. Yes, your vector should be easily introduced. That is, transformation should be easy. Okay, if it's not entering easily into the host cell, it would again, there won't be any use of the vector. It should have suitable genetic markers. So what is this genetic marker? See, now your vector having a DNA insert is being transformed into the host cell or not is very important. Okay, and we cannot use a microscope to check it. So we will be checking it out relating to some markers. Okay, so the markers should be very important so that we can easily detect which is a transformed host cell and which is a non-transformed host cell. Okay, for example, I'll tell it to you while I will be teaching the cloning and expression vectors. Okay, it's a really very easy example. It would be great if you look at the diagram and do it. Now, it should have unique restriction enzyme sites. Obviously, if you're cutting a vector, it should have a proper restriction site where you want to insert your DNA. That's quite important. So these 2 points I'll be explaining while I'll be teaching you the cloning vector just now. So these are the properties of vector, and always remember these vectors. Okay, the DNA molecules which are used as vectors, they have co-evolved with their specific natural host species. Okay, hence they are adapted to function well in them and in their closely related species. Therefore, whenever you choose a vector, it should largely depend on the host species into which you are inserting the DNA or you're cloning the gene. Okay, so always what happens is the naturally occurring vectors, they do not have all the required functions. So therefore, when you are creating a vector, you have to join few segments and perform specific functions. Okay, so let's learn about more about vectors. Here there are two types of vectors. One is called a cloning vector and one is called an expression vector. So let's learn one by one. Now, what's this cloning vector? When your main motive of insertion is to get multiple copies, then you use a cloning vector. But at the same time, when your main motive is for the expression of a certain protein or a product, then you use an expression vector. Okay, so now we are clear with cloning vector and expression vector. Let's learn more. Now, whenever you are using a cloning vector, this cloning vector should have origin of replication. This is the most important thing. Okay, see, a vector should have origin of replication because it should replicate of its own. Okay, this is the most important thing. Well, then ampicillin resistance, tetracycline resistance. These are selectable genetic markers. Do use it? How do we work on that? See, whenever you are having a particular restriction site, it should be very clear. You should be knowing it. Where is it? Now, if I am using a restriction site of BAM-H1 and STEI1, okay, and I want to insert my DNA here. So the first thing is I need a proper restriction site. Done. Now here I am inserting my DNA. That is the DNA insert. Okay, this is the region. So how do we check it now? So when you are plating this now the cell is a recombinant. This is a recombinant molecule. It has been transformed into the host. Okay, once it's transformed into the host, you will plate it, okay, to know whether the transformation is correct or not. Now, if you plate it in ampicillin resistance, you have plates. If you plate it in tetracycline, okay, so will this host cell grow there? No, they won't, because it makes the tetracycline gene inactivated. It makes it non-functional. So this is how it works as marker. Now at the same place, you have your host cell. So it proves that it does not grow in tetracycline resistance, means it has been transformed. If it grows in ampicillin resistance and not in tetracycline resistance. Okay, so this is how we use them as a selectable marker. And I think all the points which I have taught here are clear. It should have a selectable marker and should have unique restriction site for enzyme intentional cloning. Let's learn expression vector. So expression vector means therefore expression. That means it should have with origin of replication, should have a promoter, operator, repressor, chromosomal binding protein, restriction site, termination sequences, and obviously markers to know. So if it has all of these, then it works as an expression vector because it has to produce something. For that, it needs a promoter, it needs an operator to stop it, it needs a repressor to repress it, to terminate the reaction, it needs a termination sequence. Okay, restriction sites to cut at a particular point, markers for detection, okay, and ORI for the application. So this is how we use an expression vector. Okay, so this is your expression vector. Hope you understood cloning vector and expression vector. Now in the next class or in the next video, we will be dealing about one by one about all the types of vectors. It will be a detailed explanation about each. Plasmids, bacteriophage, cosmid, phagmid, plasmid, artificial chromosome vector, vector from other bacteria, shuttle vector, yeast, vector for animal, vector for plants. Keep in touch, check with the playlist to learn more.
This was all about today's video. Hope you liked it. For more such interesting video, please like, share and subscribe to BioDrunk. Let's meet in the next video. Bye-bye.